we are able to cover the basics of amino acids and peptides. So we know that there are 20 amino acids, okay? And there are 20 common amino acids, and they can be grouped depending on their polarity, depending on their side chains, right? Okay, so ito yun, yung ginawa natin yung last time. So depending on the R group, we are able to classify them as non-polar, neutral, okay, polar na neutral, and then meron tayong mga aromatic na neutral, okay? Then we have acidic and basic amino acids, okay? So do you have to remember them? Just be familiar na lang because it's fine with me if you use Codigo for now, okay? Given that we have very limited time, okay, in the lecture, so yeah, you can use Codigo na lang muna, okay? But you have to be familiar with the single letter names or the three letter names, so that it will be easier for you to recognize their um, properties, okay? So also we discussed no that there are proteins that are essential. Sorry, there are amino acids that are essential, meaning they need to be supplied with their diet. No? So ano yun? Yun yung WHT. Yan. William Howard Taft. Yun yung mnemonic natin dun, di ba? So William Howard Taft, milk. Oops. Fidel V. Ramos. Okay, so yan yung ating mga essential amino acids that we need to obtain from our diet. Okay, and most of pinaka-common dito na mapapansin natin is that this tryptophan and the phenylalanine. Okay, so this needs to be obtained from our diet. So primarily from animals or sometimes in plants. Okay, so nakukuha natin yun kasi our body cannot synthesize this type of amino acid, no? Especially aromatic stuff, okay? Because sometimes aromatic molecules are dangerous to our body, you know, it can be cancerous. So we have to obtain them from other sources, okay? Yun lang, okay? So yun, yun lang. So, also, ang significance din nito, sabi ko na last time, when we are cooking food, no, especially kapag yan ay sunog na, 'di ba, sabi ko, may mga polyaromatic hydrocarbons doon. Okay? So, those polyaromatic hydrocarbons, they might came from these aromatic amino acids, okay? Because the benzene rings can detach when the molecule is decomposing, and when you have many polyaromatic hydrocarbons in your food, it's toxic. No, it can lead to something like colon cancer, okay, and any other types of cancer. But the common one is colon cancer. Okay, so kaya yung mga may hilig sa ihaw, just be cautious, no? Huwag masyadong sunog. Okay, so yun. Okay lang yung ikaw makasunog ng food, pero wag mo kainin yung sunog na food, okay? Gano'n ako sa bahay, 'pag nagluluto ako sunog parati. Pero yun, hindi ko kinakain because I know that's dangerous, okay? So yun lang, and also we are able to have an overview on the structure of our amino acids. So our amino acids are typically in the L formation or the L isomer than the D, okay? So remember that D is the common stereoisomer for sugars, but for amino acids, it's L na. Bakit? Hindi convincing yung answer natin, pero yun na yun. Due to evolution. So we don't know why, pero yeah. Based on evolutionary ano na lang, evidence na yung L yung mas predominant compared to the D. Okay? And that is in sharp contrast with our carbohydrates. Okay? So, ayun. And then, we also knew that among all those amino acids, proline is secondary. Okay? Given that yung kanyang amino group, no, yung N group ay secondary amine. Okay? So, ibig sabihin, nakaloop. Yun, nakaloop. Okay? And that will have a significant effect later on when it comes to the structure of the protein. Also, depending on the pH, yan, nagbabago yung charges ng ating R groups ng amino acid, okay? So, depending on the pKa values, yun yung unang mag-dissociate na or not. Okay. When it comes to a point wherein the net charge of the molecule is zero, that is called the sweeter ionic species. And at that sweeter ionic species, no, the protein tends to be insoluble in water. So that technique is used by food manufacturers in obtaining some proteins from our daily produce, no, such as milk, no. So yung milk, gawin mo lang acidic yan, you will be able to get casein, which are commonly sold as your whey proteins. Okay, so yun, so legit yun, protein yun. Okay, so here are some reactions of the other other amino acids. The cysteine, ito yung dahilan bakit naging curly yung hair natin. Okay, and bakit tayo magre-rebond. Okay. And then this amino acids, ito yung mga precursor in formation of our hormones, no? So yan, ng mga neurotransmitters, yan. Okay, and other amino acids, yan. So, nakakwentaan ko naman yan last time. And then when it comes to peptide bonds, no, this is formed by condensation of two amino acids, no, wherein the carboxylic group, no, and the amino group of the other amino acid condenses. Okay, so when they say they condense, they form water at the end of the reaction. Okay, and peptide bond is characterized by a partial double bond character as the bond is resonating, no? The electron is resonating. So it's kind of strong, okay? Kasi kung hindi matibay yan, siguro tunaw na tayo. Okay, init-init pa naman sa Pilipinas. So our peptide chains are usually written from the N terminal to the C terminal, and they are all connected by a peptide linkages. Okay, so yan, so that's all that we had discussed last time, diba? So now let's talk about proteins na, okay, because we have basic knowledge on the monomers of proteins, which is the amino acid. Okay, so now let's talk about proteins. Okay, so protein is the polymer of our amino acids. Okay, so, protein serves many function, okay? So, hindi lang siya yung yung pampamuscle, no? Katulad ng kino-commercial ng mga naggi-gym lang sa buhay, no? Ni Rendon. Okay? Hindi lang siya about muscles. Marami siyang function. Okay? So, for example, proteins are used for structural purposes, okay? So, meron tayong tinatawag ng proteoglycans, okay, which are proteins with sugars that are used in connective tissues such as our joints, okay, our skin, okay. So, gawa yan sa collagen. Okay? So, kapag may collagen yan, matibay mukha mo, okay. So, ibig sabihin, hindi ka agad kukulubot, okay? Hindi ka agad mag-age, no? So, yeah, legit yung binibenta sa Watsons na collagen, no? Okay. And then keratin. So, keratin, ito naman yung ginagamit for, let's say, our hair or our fingernails. So, yan. Yan yung mga proteins na nandun. Okay? Even the bone, hair, nails. Yan. And, ayun. So, alam natin to. We are somehow familiar with this one. And another purpose of protein is to serve as catalyst. Okay? So, pag tinabi natin proteins that are catalysts, we are actually referring to what's called an enzyme. Okay? So, enzymes are biological catalysts and are made of amino acids. Okay? So, basically, protein lang din sila like other proteins in our body. But, instead of being structural, yung purpose niya, ang purpose niya is to react, to help other molecules react. Okay? And virtually all our body processes needs catalysts, no, enzymes. Okay, so for example, the glycolysis itself, no, it needs ilang enzymes ba kailangan? So I think 9 enzymes ang kailangan just to end the glycolysis, if I'm not mistaken. So I think that's 9. Okay, so you have the hexokinase, no, the transferases, the isomerases, so lahat ng 'yon ginagamit yon for those roles in the body. Okay, and the proteins that serve as catalysts will be a topic for the next discussion. Okay, so yun. So hindi lang siya muscle, no? Even our catalysts in the body are primarily made of proteins. Okay, also sa movement, ito na yung mga muscle natin. Okay, so our smooth and smooth muscles and mga sa puso, ayan. So, gawa-gawa din sila sa mga proteins, okay? Such as myosin and actin, okay? Also, proteins are used for transport. Okay, so transport, one example of which is hemoglobin. Okay, so hemoglobin, it circulates oxygen in the body and releases carbon dioxide produced by cellular respiration. Okay, so after ma-breakdown ng food mo sa cells natin, ayun, ilalabas niya yung carbon dioxide from the cell papunta sa lungs natin where we exhale it. Okay? And then when we inhale, papasok na uli yung oxygen and it will transport all the oxygens to the body. Okay? To continue the metabolic processes. Okay? So that's one of the best example. So hemoglobin. Okay, so proteins are also used for hormones. Okay, katulad nga nandun sa previous slides, no? Our oxytocin, and mamaya may examples pa tayo about this one. Okay, so yun. And protection, so proteins, ginagamit din yan for blood clotting, for example. And storage, protein, ano ba yung pwedeng example dito? Let's say, mga lipoproteins, okay? So, ginagamit siya sa transport and sometimes ginagamit din siya for storage, okay? And then, lastly, for gene regulation, okay? So, yan. So, yan lang. Hindi naman kailangan kapisaduhin yan. But, those are some of the many functions of proteins, okay? Yan. So, depending on the level of structure of your protein, it could have several implications kung ano yung pwede niya gawin. Okay? So, our levels of structure of proteins are characterized into four levels: the primary, secondary, tertiary, and the quaternary. Okay? Each of them focuses on a different point of view for each protein. Okay? So let's talk about the primary muna. Okay, so when you say primary structure, we are actually referring to the individual links, in the individual chain of amino acid. Okay, so dito natin makikita yung pagkakasunod-sunod ng amino acid. For example, methionine, cysteine, no? Yung uh, histidine. Yung mga sequence nun. Okay? So, yun yung primary structure, yung sequence of amino acid. And this is what we use actually in determining if meron tayong COVID-19. Okay? So, that's what our molecular biologists, medical technologists do. No? They collect samples of proteins in the body and then they test it against the record or the database for the SARS-CoV-2. Okay? So for example, in this protein, the LXPR, LPXR pala. So this protein, LPXR of Salmonella. Okay, so these letters here, ayan, so all those letters, they all correspond to, ayan, the first level of protein structure. That is the primary structure. Okay, so yan. So mamaya, balikan uli natin yan. Okay. So when we talk about the primary structure, again, we are referring to the individual chain, okay, the individual components of the chain. Next one is the secondary structure. Okay. In the secondary structure, we see our amino acid chain, no, forming some form of some form of conformation. Okay, so pag sinabi natin secondary structure, some of them may be alpha helical like our DNA. Some of them can do beta sheets. Okay, so minsan flat sila. Okay. Later marami tayong picture for that. And then kapag tertiary structure, we are looking at the overall view of the monomeric protein. So parang nag-zoom out na tayo. We see protein as a whole, and then pag quaternary, we see protein as packets, okay, of different protein chains, okay? So yun, sa bale, ang pinaka-simplest point of view is the primary, we see the sequences. Secondary, medyo mag-zoom out ka. Makita mo, they form alpha helix, beta sheets, okay? And then pag tertiary, mag-zoom out ka uli, you look at the protein as a whole, okay? Its monomeric structure, and then the quaternary is the combination of all those proteins, okay? So yun yung quaternary structure natin. Okay, so as I said, primary structure are the sequence of amino acids in the polypeptide chain, and All of them are derived from the 20 common amino acids. Okay, so yeah. So for example, you have a 20 carbon chain length, so the possible combination is 400, and if you have 20 by 20 carbon chain length, ayan, so the tripeptide chain, okay, is is possible to form ano, tawag dito, ano ta? 8,000. Okay, so it can form 8,000 patterns or sequences. Okay, so bale the number of sequence that we can make out of the number of protein chains that we can have, is 20 raised to n. Okay, so suppose natin you have a pentapeptide sequence. Teka lang, lobat ako. Okay. So suppose you have a pentapeptide sequence. So 5 yung ano, amino acid residues mo. So bilangin natin ilang combination pwede magawa. So 20 raised to 5. 3.2 million. Okay, so 3.2 million combination yung pwede natin magawa. So imagine the possibilities. No? Sobrang daming pwedeng pagdugtongin na amino acid. So that's why life is diverse. Okay? Hindi lang tayo single-celled organism tulad ng dati. No? Iba't ibang organism na tayo. No? May sariling uh, may sarili ng logical reasoning, no? May sarili ng ginagawa sa buhay. So yeah, so that's the reason why life is so complex, no? There are so many possibilities na pwede mo dugtungin yung ating mga amino acids. Okay? So yan. Oopsie. Okay. So in this picture here, we have the insulin. Okay, the insulin. Saan to ginagamit? In regulating sugar levels in the body. Okay. So, insulin, ito yun, dalawa itsura niya. So, dalawang chain yan, connected by the cysteine linkage, okay? So, yan. So, pikita mo yung cysteine dyan, they form the disulfide bridges, okay? So, ang papansin natin is that, ayun, those individual circles there are the amino acid chain, okay? And so, glycine, isoleucine, valine, uh, glutamic acid, glutamine, cysteine. Okay, so nakikita nyo, in the primary level of amino acid structure, you can see the individual amino acid residues. Okay? Ayan. Okay? So, yan. Hindi nyo naman kailangan kabisaduhin yan. Ang basta ang kailangan nyo lang alamin is that for primary level, yan, nakikita mo yung amino acid sequences. Okay? So, how important yung ating amino acid sequence? Okay? So, a minor change in the amino acid sequence can cause a big difference. Okay? Kasi proteins follow this format called uh, structure follows function. Okay? So, depending on the structure of your proteins, magbabago yung function niya. So, for example, asan ba yung example nun? Okay, so for example, ito. So, the vasopressin and the oxytocin, okay? So, both are nonapeptides, but they have different biological functions. For example, kapag vasopressin, ginagamit yan as antidiuretic hormone. So, ibig sabihin, hindi yan, pampahinto yan ng ihi, no? And then, kapag oxytocin naman, this is used to affect the contraction during childbirth, okay? So, as you can see, siya, non-peptide, pero just look, saan lang sila nagbago? On the second and on the seventh amino acid sequence. Okay? So, since doon lang sila nagbago, it doesn't seem na malaki yung effect nun, but it is, no? It causes great effect, no? Okay? So, yun. So, ibig sabihin, palitan mo lang yung amino acid sequence mo, palitan mo lang yung primary level of structure mo, your protein function will change very dramatically. Okay? So, to give you much understandable context, no? Punta tayo sa first amino acid, the first mutation of COVID-19, which is the D614G. Okay. So, my former biology students na ayun, siguro familiar pa sila dito kasi by the time na tinuturo ko tong Gen Chem sa kanila, yung Org Chem, okay. So, ito pa lang yung mutation na meron sa mundo. Okay, that's D614G. So, the D614G, this is the first mutation of our COVID-19, wherein the 614th sequence, the D, which is the aspartic acid, was converted into G, the glycine. Isang amino acid lang nagbago. Okay. What happened? Kumalat lalo ang COVID. Kumalat lalo. Okay. So probably ang dahilan nun is due to ano, yung isang amino acid sequence change na yun, it resulted to much more spike proteins. Okay? So, yun yung significance ng ating, ang dami naman nitong ganito. Ayan. So, yun yung significance ng primary structure of amino acid. Baguhin mo lang isang sequence, magbabago na yung function ng protein mo. Okay? So, for example, in this PowerPoint, we have the vasopressin, which is used for antidiuretic purposes, and the oxytocin, ginagamit to for childbirth. Okay? So, two things are very different, di ba? Pero ang nagbago lang sa kanya, dalawang residue. In the context of pandemic, ang nagbago lang dito is one amino acid residue from D, naging G. But look at what happened now. By the time this arose, no, by the time na na-detect to sa, I think this was detected in Europe, if I'm not mistaken, Germany. So, yun, kumalat no, ang COVID. No? So, ito yung nag-cause ng mass matinding spike no, ng COVID cases late 2020. Okay? Bago pa yung February 2020 na spike. Okay? So, yun. So, in the Philippine context, alam natin yung P3 variant. P3 variant ng COVID-19. So, that is just alteration also of the primary level of structure. Okay. Sequence. Okay. And so let's take a look at the P3 variant. Okay, so wala tayong makuha masyadong data kasi sa Philippines siya. And eto. So I hope that we can see some info here. Okay, so ayan. Basahin na lang natin. So, the 3 amino acid deletion at the positions 141 to 143. Okay, LGV, leucine, glycine, valine, nawala sila. And by just deleting that, ayun, karoon tayo ng panibagong variant. Okay? So, yun yung significance ng ating primary sequence of proteins. Okay? Okay. So, also in the sperm whale, okay, sperm whale ng uh, sperm whale na. Alam ko mayroong protein dun eh. Na very analogous with humans pero iba yung later hanapin ko na lang siya habang inaalala ko. Okay, so I forgot to search for that in advance. Pero yan. So as you can see, by just changing one amino acid residue, malaking effect niya when it comes to the structure. Okay? So yun. So ganun katindi yung significance ng primary structure. Okay? So kung tanungin kayo, okay, especially medical biology, karamihan sa inyo, right? Okay. So, kunwari, tanungin kayo ng mga kamag-anak nyong hindi naniniwala sa mga variant, no? So, bakit sasabihin nila, ay, yung vaccine yan yung nagkakos ng variant? No, hindi ganun. Okay? Sabihin nyo sa kanila is that the reason why variants exist is probably due to amino acid, yung changes sa primary level ng amino acid. Okay? So, since nagbabago yon over time, that is bounded by Mendelian, hindi pala Mendelian, Darwinian theory, okay? Survival of the fittest lang yan, maski sa proteins, okay? So, gagawin ng mga proteins natin, mag-adjust sila, mag-delete sila, mag-add sila, para mas mag-survive sila, diba? Ganoon naman yung buhay survival of the fittest. So, in order for you to survive, proteins must also evolve. Okay, that's why until now may COVID-19. So, in order for us, not to produce any more deadly variants like the Delta, okay, kailangan natin is maging vaccinated. Okay, kasi when we are fully vaccinated, okay, most of the world gets vaccinated. That is the time when this viruses won't hindi na siya magiging sikat. Hindi na siya magiging exposed sa environment. So ibig sabihin mamamatay na lang siya. Okay? But according to some scientists, it appears that COVID is there We are going to live with COVID in the future. Magiging parang flu na lang yan, okay? But before that happens, sana wala na mamatay, okay? So, paano gagawin yon? Edi magpa-vaccine tayo, okay? So that's the best, that's the most effective way of not getting killed by the virus, okay? Kasi COVID is there to live with us talaga, okay? Kasi coronavirus din yan eh, okay? Just like our other form of viruses, no? Mga flu natin, ayan. Coronaviruses din yan eh. Kaya yun, this pandemic will never end for sure. Pero ayun, I hope that before mangyari yon, no, sana our body gets immunized na. Okay, masanay na tayo sa COVID para maging regular flu na lang yan. Okay, so yun yung nababasa ko from other papers. Okay, so ganun yung trend natin. We will live with the virus. So yan, so get vaccinated, guys. Okay. And then next one, so we're done with the primary structure. Now let's talk about the secondary structure. Okay, the secondary structure of the protein can be inferred from its primary structure. Okay, so pwede mo malaman yung secondary structure na pwede niya gawin kung alam mo yung primary structure. Okay, so commonly naman, dalawa lang yung uri ng secondary structure. We have the alpha helix and the beta pleated sheets. Okay, so kapag sinabing alpha helix, ito yung parang sa DNA. Okay? Nag-wind-wind siya. And kapag beta pleated, ito yung flat. Okay? So, yan. So, mamaya, sabihin ko sa inyo paano nyo malalaman kung anong type ng secondary structure yung makikita nyo sa protein. Okay? Sige. So, next slide muna tayo. Okay. So, the secondary structure of protein is usually governed by hydrogen bonding. Okay? And the hydrogen bonding can exist, no? In our amino acids, okay? So, katulad ng amine groups, no? Ng ating alpha amino groups, and then yung ating carboxylic groups, okay? So, they can form hydrogen bonding. So, that's why the secondary structure gets stable, okay? So, yun. And also, the rotation of the bond can also happen. Ito. So, dito nangyayari yung rotation of the bond. So, remember, this is the peptide linkage. Okay? However, the sp3 hybridized carbon, this can freely rotate. So, ibig sabihin, pwede nga siya mag-flip-flip. Okay? So, although the peptide linkage is fixed, this sp3 hybridized carbon can rotate. Okay? So, that causes the 3D conformation of our proteins. Okay? So, in the alpha helix, ganito yung itsura niya, just like your DNA. So, the peptide chain, ito yon. So, nakikita nyo, it is fixed, no? It is placed along the line, okay? Along the ribbon of the helix. Pero, yung nagro-rotate dyan is the sp3 hybridized carbon, such as this one, okay? So, sila yung nagro-rotate. That causes alpha helix turns, okay? So, what's the characteristic of our alpha helix? Okay, so the characteristic of our alpha helix is this, no? So there are apparently 3.6 amino acid per turn of the helix. Okay, so in this turn, so ilang amino acid makikita natin? So probably ito ay 1, this is 2, this is 3, then ito yung 3.6 na. Kasama pa siya dun sa other eh. Okay, other ring. Okay, so bale, for every turn in our alpha helix, there's an approximately 3.6 amino acid residue per turn. Okay, and as you can see, 6 atoms of each peptide bond line on the same plane. So ito yon yung pinapatukuyan natin. So these are the 6 atoms. Syempre may kadugtong pa yan dito. Okay, so the 6 atoms of our peptide chain lies in the plane. So yun lang yung sabi dyan. And then the NH group points at the same direction. The C double bond O groups also point on the opposite direction. Okay, so tignan natin. So as you can see, these are the CO groups. The C double bond O ng ating amide group. So they are pointing downwards. Okay, so ibig sabihin, the C terminal are pointing downwards. But look at the N terminal. The N are pointing upwards. Okay, so ano yung mangyayari kapag ganun? So that causes our alpha helix structure to form macrodipole. Okay, so when you say macrodipole, para siyang isang malaking magnet, no? wherein the N groups are all pointing upwards, the carbonyl groups are all pointing downwards. So, on the top portion, dito yung partial positive side ng ating molecule. And on the bottom portion, dito yung partial negative side ng ating molecule. Okay? So, yan. That is caused by the different orientation of our carbonyl and the amino groups. Okay? So, yan. And keep in mind that the carbonyl are hydrogen bonded to the amino group. Okay, so look at this one. So this is our carbonyl from this peptide chain. It is bonded to this amino group no, via hydrogen bonding. So that's why it is stable. No? The helix is stabilized um, by the hydrogen bonding. Okay, so yun yung motif niya. No? Lahat ng carbonyl pointing downwards. Okay, tapos, lahat ng amino groups pointing upwards. Okay? And the one that stabilizes them, and something like that. So, the one that stabilizes them is the amino group of the other residue. Okay? And then, all our groups are pointing outward the helix. Okay? So, all the R groups, they are pointing outwards. Okay? So, I think yan yung mga yellow. Okay? So, sila yung mga naka-point outwards. So, ibig sabihin, to give you an itsura, kung to give you an idea kung ano itsura niyan, kuha tayo ng ito. May alpha helix ba dito? Can you see an alpha helix? Ayan. So this one is an alpha helix portion. Okay? Oopsie. So this portion here is the alpha helix. Ayan na. Okay? Nakikita niyo yan? So ayan, nakaparing siya. Ayun. So, that is the alpha helix. Okay? So, mag-zoom in tayo. Okay. So, this is the R group of this amino acid sequence. Ano ba yung sinelect ko? Ang liit kasi ng screen na to eh. Liit ng second screen ko eh. So, that, yeah. Yes. Okay, so asparagine daw to, yung clinic ko na to. Okay, so as you can see, for the asparagine, all the R groups are pointing outwards. Ayan na. Oops. Zoom in ulit tayo. Ito, ibang residue naman to. So, this is the R group of this amino acid. This is the R group of the amino acid na yon. Ay, nasa likod siya. So, ito yung R group ng amino acid na to. So, probably this is valine kasi letter V. Ito, I don't know what's this. Kasi nasa side view. Okay. So, ayan. So, as you can see, all the R groups are pointing outwards. So, probably this is 1, 2. Glutamic acid to. So, ayan. So, ganun. All the R groups are pointing outwards. And look at the inside of the helical structure. Nakikita nyo yung amide group pa yun. Flat siya doon. These N groups, the amino group, is hydrogen bonded to the carboxyl group of the other residue. Okay? So, ito yung sinasabi nating hydrogen bonding kanina. So, this is the N of the peptide chain, the NH. Ito yung carbonyl group ng another peptide chain. Okay? Nung time namin, di namin alam to kaya di namin ma-visualize. Kinakabisado lang namin. Yung pala, pwede naman pala gumamit ng ganitong website. Okay? So, ayan. So, let's take a look at this portion. Ayan. So, as you can see, all the R groups are pointing outwards. And our carbonyls, they are pointing in one direction. And the amino groups are pointing in the other direction. And the carbonyl and the amino groups are hydrogen bonded to each other. Okay? So, that stabilizes this alpha helix structure. Okay? So, ayan. Mamaya marami pa tayong example. Kinuha ko lang yung picture na yan kasi obvious na obvious yung alpha helix and the beta sheets. Okay? So again, ito pala yung tanong ko. Ito yung hindi ko nasagot kanina. Paano mo sir malalaman kung mag-alpha helix yan? Okay? So malalaman mo if that is an alpha helix if every after 3 residues, same yung polarity ng iyong amino acid. Okay? So every after 3 amino acid residues, same yung amino acid sequence mo. Okay? So apparently hindi ko siya ma-view na maayos dito because masyadong maliit ang screen. Anyway, mamaya na lang. Basta yun yung idea, no? For example, in your primary structure, may makita ka valine, then after the three residues, you have another valine o kaya isoleucine. So probably, it can form alpha helix. Okay? So yun yung aming technique to determine if that's alpha helix. Every after three residues, same yung polarity niya. Okay? So yun. Okay? So yan, ano to? This just showed the macro dipole. No? All the carbonyl groups are pointing in one direction. All the amino groups are pointing on the other direction. And just like what we saw kanina, this carbonyl and this amino group are hydrogen bonded to each other. Okay. So there are several factors that can disrupt the alpha helix. Okay. So although they are hydrogen bonded to each other, there are some residues that can destroy it. One of the residues that can destroy the alpha amino acid is the alpha helix rather is the proline. Okay. So what's the thing with proline? Okay. So proline is a secondary amino acid. So ibig sabihin yun, yung kanyang configuration, medyo hindi siya akma for for alpha helix. Okay? So sabi diyan, proline causes bend because of the restricted rotation due to its cyclic structure, and its alpha amino group has no NH for hydrogen bonding. Okay? So katulad ng pinakita ko kanina, okay? So if you have a proline here, meron ba tayong proline? There's so many amino acids I cannot check kung may proline. Ito. So this is a proline part. Okay. Yeah. So this is a proline part. The histidine yan. Ito. So this is the proline part. Now, look at what happened at this region. Okay. Yung ribbon na ganun, look at what happened. So this is the proline part. Tignan nyo. Wala na. Naputol na yung alpha helix. Okay. So the alpha helix gets disrupted whenever we have proline high residues. Okay. Kasi due to its configuration, it cannot stabilize the CO and the NH bond, no? Kasi wala siyang hydrogen for H bonding. So that's why it gets disrupted. Okay. So tignan nyo, for the rest of the part of the molecule, the alpha helix is okay, but on this region, disrupted na. Okay. So yan, dito yan na banda. Dito yung proline kanina. Okay. So yun. So that's one of the residues that can affect the 3D structure of our amino acid, the proline. Okay. Number one, and number two, other factors that can disrupt our alpha helix is that if ever we have residues that have strong electrostatic repulsion, especially we are pertaining to charged R groups such as histidine, arginine, lysine, glutamine, aspartic acid. No? So kung magkakatabi sila, instead of them forming hydrogen bond, they can actually repel each other because they possess positive charge or negative charge R groups. So yeah, they can destroy the linkage. And also, due to steric crowding as well, okay, so even though non-polar to, they can also cause bending in the alpha helix, no? Such as for valine, yung letter V niya, okay, so pwede tumama yun with the other R groups, so mabibend niya yun, okay, due to electrostatic repulsion. Okay, so yan. So those are the factors that contributes to the alpha helix, and yung nakita natin in our protein, no, the LXPR protein ng salmonella, yun. Kita nyo yan. Okay. So yan. So again, what are the things that you have to remember for alpha helix? For alpha helix, there are 3.6 amino acid residue per turn. Okay. And what else now? The polarity of every residues, no, every after three amino acids, no, they are the same. Okay. So pakita ko sa inyo yung prob set ko sa grad school. Yung kasi maganda eh. Teka lang, kunin ko lang yung prob set ko sa grad school. Kasi yan yung mismong lesson namin doon. Okay. Now, don't worry, di naman kayo bibigyan na ito. As for example, ito, we are asked here, for the protein, okay, paano mo daw nasabi na alpha helix yan? Kasi look at this. No? So for example, in this region, amino acid residue 39 to 58, okay? So every after 3 amino acid, there is a similar, okay, polarity among the residues. Okay? Ayan. And so for example, this is uh, ito na lang. So, this is alanine, except for this one, and then valine. Okay. So, yun, mapapansin mo, may consistency kahit paano sa ating amino acid residue, and that causes it to form the alpha helix. Okay. So, this is phenylalanine, glycine, alanine, glycine. Okay. So, these are all non-polar, diba? So, yan. Yan. So these are all non-polar, that's why this region forms alpha helix. Okay? So yun. So yun yung technique, no? Paano mo malalaman kung siya ay alpha helix. So every after 3 residues, you have your similar polarity, and ayan, they form alpha helix. So next secondary structure of the protein is the beta pleated sheet. Okay. So pag beta pleated sheet, ibig sabihin nun, flat. Okay. So yun yung nakikita nyo kanina sa ating pictures dito. Okay. So the beta pleated sheet are these regions here. Ayan, yung flat na yan. Ganda, no? So yan. Ayan, so these are the beta pleated sheets. Okay. So sila yung mga flat part ng molecule. Okay. So ano meron doon? Of course, they can also do hydrogen bonding, no? Pero iba yung motive nila. Okay? So yun, puntahan natin yung mga motive. Okay? So in the section of beta pleated sheet, okay? Okay. Sorry. Hindi kayo, ha? So in the section of your beta pleated sheet, okay? So your amino acid residues are lying on the same plane, kaya flat siya. Okay? And also, the carbonyl and the NH group of the adjacent chains, no, point towards each other. Okay. For example, this is one chain, this is the other chain. Their NH bonding, no, yung hydrogen bonding nila dito, they exist between them so that they can stabilize the beta pleated sheet. So let's take a look at this structure here. This is a wonderful example. Ang ganda niya, no? So tignan niya, so this is one chain, this is the other chain. Nakikita nyo yung broken line. So that is the hydrogen bonding between this chain and the other chain. Okay? So imbes na perpendicular yung perpendicular sa R group yung ating hydrogen bonding like alpha amino acids. Sorry, the alpha helix pala. Okay. So dito naman, instead nasa iisang chain yung hydrogen bonding, dito nasa magkabila ang chain. Okay. Kapag ganun siya. Yan. So this is one chain, this is the other chain, and then the hydrogen bondings exist between them. Okay. And that stabilizes the beta pleated sheets. Okay. Ano pa kailangan niya sabihin? All the R groups are alternating. Okay. So alternating din yung R groups. Okay. So ito yung tanong. Sir, paano mo malalaman kung beta sheet yan? Okay. Malalaman mo kung beta sheet yan if alternating yung polarity ng ating residues. Okay? So, let me show you one example. Tinanong yan din yan dito sa prop set namin. Okay. That's one RTQ protein. Mamaya na yan. So, ito. So, tinanong sa amin, at this residues, ano yung napapansin mong pattern? So, usually, kapag yung alternating residues mo ng amino acid, if they have the same polarity, then high yung chance for them to form high yung chance nila to form the beta pleated sheets. For example, itong chain na to, siguro magkakahilera sila. So look na. So at this point, alanine yan. This region, except for the D, so valine and isoleucine, pare sila ng polarity. Okay, and then you have this, no? You have that. Okay, so yun, mapapansin mo that alternating yung polarity nila. Okay, and so yun, ayun lang. Okay, so again, compared to your alpha helix, no, na every after 3 amino acid residue, yung similar polarity. For the beta-pleated sheet, no, every after amino acid yung similar polarity na. Okay? So, nari, alanine ngayon, the next sequence, the next residue should be nonpolar as well, okay? Such as glycine, okay? So, yan. So, there are two types of beta-pleated sheet, no? We have the parallel and the anti-parallel. Okay? So, when you say parallel, same yung direction ng ating chain, okay? Ayan. For anti-parallel, opposite yung direction ng ating chain. And as you can see, in the anti-parallel strand, no, such as this one, magka-opposite yung direction nila. That's why they are called anti-parallel. As you can see, the formation of hydrogen bonding is better than in the parallel. Okay? So, mas maganda yung hydrogen bonding kapag anti-parallel compared to parallel. Okay? So, just like our alpha helices, no, sometimes hindi yan completely flat. Okay? Sometimes may mga bulges or may mga may mga curves. Okay? So, yan. For example, in the beta bulge, okay? So, kailan to nangyayari? This usually happens in the anti-parallel structure and it is an irregular, non-repetitive pattern. Okay? So, ano yung mga dahilan for that? Probably, you have your glycine. Okay? So, glycine, since ang glycine ay sp3 hybridized, okay? So, it can rotate, no? So, the rotation of the glycine can cause these turns, okay? Yung mga bulges na yun, or yung mga turns, okay? So, yun. Balik ulit tayo sa ating protein na nandito. Tignan natin kung may glycine. Although, di ko pa na-check kung may glycine. Sa ating mga turns, meron ba tayong turns? We don't have turns dito. Wala tayong beta turns. Okay, so akala ko meron. Anyway, so yun. So, sa mga turns, usually, or sa mga bulges, no? Okay, you have your glycines there. Okay, because glycine can freely rotate. So, yeah, it can, instead na iisang straight line yan pa ganun, pwede siya mag-flip pa ganun. Okay, so, magkakaroon ka ng turns. Also, prolines are encountered in turns. Okay, other than glycine, you also have proline. Okay, bakit daw? Okay, so, ang dahilan doon is that yung ating proline, dalawa kasi yan eh. You can either have the cis or the trans proline, no? So, if you have your cis proline, ganito yung itsura nan. Okay. So, for example, this is your amino acid residue. Ayan. So, it can actually cause your, chain to fold like that, okay? To turn like that, okay? So, if yung ating proline ay naka-cis, no, then it is the time wherein you can see turns in your beta sheet. Okay? Kapag trans naman, okay, so wala dyan. No? Most likely is linear pa rin yan. Okay? Most likely helical yan. Okay? So, ganun. Okay. Ayun ang dahilan bakit may proline din tayo for the turns. No? If you have a cis proline, it can actually conform to the cis configuration and that causes your beta sheet to fold like that. Okay? Now, gaganon yung ating beta sheets. Okay? And this is common for anti-parallel. Okay? For the anti-parallel beta sheets. Okay? Usually may cis pagdating dun sa turn na yan. Okay? Ayun lang. Okay? So, sometimes there are super secondary structures. We call them the motif. Okay, these are the combination of the alpha and the beta sections of our protein. Example niya is the beta-alpha-beta unit, the alpha-alpha unit, the beta meander, the Greek key, and the beta barrel. Okay, so ano yung tsura nila? Okay, so ganito yung tsura nila. So for the beta-alpha-beta pattern, okay, so you have your beta sheet here, and you have your another beta sheet there, and they are connected by the alpha helix. Okay. So that's the beta alpha beta motif. Okay. So I'll give you an example of that. Okay. So let me just look it up. No? I think yung 1RTQ yun. Ata magaganito kaya no. Anyway. Yung upuan ko yung tumunog, hindi ako yun. Defensive lang. Okay. Ayan. So, in this structure here, we can see a beta alpha beta conformation. Asan yan? This is 1-RTQ. Anong protein to? This is the aminopeptidase of Aeromonas proteolytica. Okay. I don't know what is that. Pero alam niyo yan. Biologist kayo eh. Okay. Ang hirap naman tumingin dito. Maliit kasi yung screen. Asan kaya yun? This one. Okay. So this is beta sheet. Okay, so this region is a beta sheet and it is connected to an alpha. To an alpha. And then it is connected back again to this beta region. Kita nyo yan? Ang hirap kasi tignan eh. Ang dami, ang gulo eh. Pero yan, that's the beta alpha beta. Oopsie. Ang dami nangyayari. So that's the beta alpha beta conformation. So for example, this is the beta sheet region. Beta sheet. Then pagdating dito, kukoil lang siya. Then you have your alpha. Then pagdating doon, babalik siya to this beta. Okay? So that is the beta alpha beta motif. Okay? So beta flat, magko-coil, then balik ulit sa flat. Okay? So yan. Have you tried checking out this website, no? Kahit magganong-ganong ka lang, no? Para kunwari nag-aaral ka sa bahay. Wow. Pero hindi mo alam kung ano ginagawa mo. That's fine. And try nyo lang. Kasi nakikita nyo yung mga secondary structure ng proteins. Okay? So this is the alpha helix. This is the beta sheet. Okay? And then saan yan nakadugtong? Hanapin mo. Saan yan nakadugtong? Sa alpha na naman. Okay. So anyway. So ganun. So that's the beta alpha beta turn kanina. Pinakita ko. Flat, magko-coil, then flat ulit. Okay? So that's one of the super secondary structure, the beta alpha beta unit. Sometimes you have the alpha alpha unit. Okay? So I think yung previous na pinakita ko, this one from the Salmonella, I think meron tong alpha alpha eh. Yeah, so this is an alpha connected to an alpha. Okay? So although maliit lang yung alpha niya. Okay, so that's the alpha alpha secondary motif. Okay? And then what's next? Ano to? So this is the beta meander. So kapag beta meander daw, pag ganun-ganun. Okay? Pa zigzag yung ating beta sheets. I don't have an idea kung anong pwede kong gawing example dyan. Pero yan, basta may ganun daw, beta meander. Okay? And then you have your D. This is the Greek key. Okay, the Greek key. Ayan. Which is, bakit daw ganyan tinawag na Greek key? Is because it follows the traditional patterns in Greek porcelains na porcelain nabubulol na. And so yung mga Greek porcelains daw, may mga ganun daw na structures daw. May mga ganun siyang picture. So that's why yan yung tawag sa kanya. Okay? And of course, the last one, okay, yung dahilan bakit yun yung pinakita ko sa inyo, the beta barrel. So this is the beta barrel and this is from Clostridium. Basta yun. Hindi ko na mabasa. Malabo mata. Okay. So yan. So that is the beta barrel. Okay. So these are primarily used by bacteria. Okay. So such as this one. This is from Salmonella. Nakikita nyo yung beta barrel. Obvious naman, di ba? Ayan. So you have your beta sheet. They are somehow revolving around this center na medyo hollow. Okay. So yan yung beta barrel natin. Okay. Para siyang PVC pipe. Alam mo yung yung hose sa bahay. Diba, parang pag ganun sila, di ba? Okay. So that's your beta barrel. So parang silang naka-cross then paikot. Okay. So ano yung purpose na itong beta barrel? So commonly, the beta barrels are used by different bacteria, okay, in order to gain access to the cell. Okay. For example, you have your cell surface here. May cell surface ka dito. Gagawin nila yung mga bacteria, itutusok nila yung kanilang beta barrel and then doon nakakapasok yung yung mga contents nila. papasok sa cell. So yeah, they are used primarily for transport. Okay? So gagawin nila, tutusok nila yan sa cell, and then dito sa loob ng barrel, doon mag-flow yung kung ano mang gusto niya ipasok sa cell. Okay? Something like that yung kanyang function. Okay? So yan. And nung nakita ko tong protein na to, sabi ko, ang galing. Okay? So try natin ni Google to. Pakita ko sa inyo yung kanyang sa protein data bank natin. Okay? Para mas makita natin yung para maikot-ikot natin siya. Beta barrel. Anyway, wala, hindi ko makita yung mga exact proteins niyan. Pero ito na lang. So this is another protein. Okay, so this is the N-terminal beta barrel domain of Pseudomonas aeruginosa. So yan. Ayan. So ayan, wow. Wala lang. Wow lang. So that's the beta barrel. So they are primarily used by bacteria. So itutusok nila yan sa cell. Doon nila ipapasok yung contents nila. Okay? So yan. Okay? So yan. Yan, gawin nyo sa bahay after this class, magganyan-ganyan kayo sa bahay. No? Wala lang. Ako na rin na masipag. Okay? So yan. So those are the motif, no, for the secondary structure. And then sometimes, di ba may nakikita kayo kanina, random coils. Okay? So random coils. So we have random coils then. Okay? So andito yon. For example, yung ito, asan yung random coil dito? Ito. So kung may nakikita na parang parang gulo-gulo lang, wala siyang secondary structure. For example, this region here. It's called a random coil. Okay? So the primary reason bakit dyan siguro may random coil is dito concentrated yung mga charged amino acids natin. Okay? So our charged amino acids are primarily concentrated there. So that's why di sila makagawa ng alpha helix or beta sheet. Okay? So ganyan sila. Oops, gulo. Gulo-gulo sila. Okay? Ayan, tignan nyo. So this is ano to? This is aspartic acid. Okay. Okay. So aspartic acid yan. And then ay, hindi ko na makita. Basta yan. So most likely in this residues here, you have your charged amino acid residues. Kaya hindi sila makagawa ng magandang coil. Okay. So in this picture here, meron ba tayong ano kaya to? Hindi ko alam kung ano yan. But meron dyan. Okay. So anyway. So yan. So yun yung random coils natin. So they are primarily composed of charged amino acid. Kaya hindi sila makapag-coil. Okay. So yan, secondary structure pa rin tayo. So many globular protein. When you say globular protein, sila yung mga naka-globules, nakapabilog. Okay? Contain all the three kinds of secondary structure: the alpha helix, the beta pleated sheet, and the random coil. Okay? So this is one example, the enzyme carboxypeptidase. Okay? So in the enzyme carboxypeptidase, you can see the alpha helix here, the beta sheets, right, the beta pleated sheets, and the random coil, which is wala silang defined secondary structure. Okay? So yun. Okay? So in the collagen triple helix, okay? So in the collagen naman natin, so it is important to note that the collagen appears to have a triple helix, no? So hindi lang siya single helix, no? So naka-triple helix yan. So due to its triple helix, no, mas na-maximize nila yung hydrogen bonding. Okay? So the reason, yun yung reason bakit matibay yung collagen. Okay? Kasi kung hindi matibay yung ating balat, siguro nagli-leak out na yung contents natin. No? But yeah, collagen is triple helix molecule yan. So yung ating mga amino acid chain dyan, naka-triple helix sila. Para silang sinulid, di ba? Yung sinulid, pag hinila mo yon, di ba tatlo yon or apat minsan? So naka-weave sila pa ganun. Okay, so parang ganun yung collagen, no? Okay, so naka-weave sila paikot sa isa't isa. So that's why they are strong, and they are used for skin in our skins. Okay, so another important thing to note for the collagen is that meron tayong mga special amino acid residues dito, like the hydroxyproline. Okay, so may proline tayo pero in the hydroxy form. Okay, so the purpose of this is to increase the hydrogen bonding. Okay, so this increases the hydrogen bonding, making collagen much stronger. Okay, then sometimes we have repeating sequences of X-progly or X-hypgly. So itong hype is the hydroxyproline, wherein X is any other residues. Okay, so ito yung repeating pattern for our collagen, making sure that the hydrogen bonding is maximized. Okay, so yun. So yun yung important thing dito. Triple helix siya, ibig sabihin maraming hydrogen bonding, and also some of the proline residues are in the form of hype or hydroxyproline, which is just a proline with extra OH, no? And that OH causes extra hydrogen bonding. Kaya matibay yung ating mukha. Okay, so yan. Some proteins are characterized also as fibrous. Okay, so they tend to form long sheets. Okay, and they are technically insoluble in water. Examples of fibrous proteins are collagen, keratin, and the cartilage. Okay, so yan yung keratin sa ating buhok. Okay, so that's entire protein thing. Okay, fibrous, ibig sabihin it forms fiber. Isang sheet lang siya. Okay, isang coil or isang sheets. Okay, so ganun. So that's the fibrous proteins. Some proteins are globular in structure, and usually globular structure proteins are found inside the body. Of course, wala kang makikita na globular sa labas ng katawan natin. Mostly, they are fibrous, okay, to form a barrier. Pero yung ating globular, these are most likely used for transfer, okay? So, sila yung ginagamit for transfer enzymes, okay, such as hemoglobin, okay? Our hexokinases, they are globular in structure, okay? So, yun. And they tend to be soluble in water. Bakit kaya sila soluble in water? The thing with globular protein is that their amino acid residues, no, yung kanilang non-polar amino acid residue, they are inside the cell, inside the protein. Okay? So nagka-clump sila together kasi yung mga non-polar nasa loob. So that means yung mga non-polar amino acid residues nasa labas. So that causes them to be soluble in water. And because the polars and the polar residues are outside the globule, yun, it can do hydrogen bonding with water, making it more possible to be transported around the body. Okay? So ayan. So these are some examples of globular protein. So this is myoglobin. Okay? So ito ay this is somehow similar with hemoglobin. Okay? So yan. So it is globular, and as you can see, may mga repeating patterns tayo dyan ng beta. Alpha sheets. Okay? Yun lang. Wala na bang ibang picture? So anyway, mamaya, pag may time pa tayo, tingin pa tayo na ibang examples. Okay? So, mamaya na lang. Next one, we have the tertiary structure. So, tapos na tayo sa secondary. So, basically for secondary, you have your alpha and beta. And then sometimes the combination of them forms super secondary structure, such as the beta-alpha-beta, the alpha-alpha, beta-meander, Greek key, and the beta barrel. Okay? So, they have certain functions, especially for bacteria, in order to transport chemicals, no? Siguro to the cell, something like that. Okay. So now let's talk about the tertiary structure. When you say tertiary structure, we are looking at the wider point of view. Okay, mas malaki na yung point of view natin ng ating protein. So this is a 3D. Okay, and ang tingin natin dito is malayuan na. Okay, so what are the factors that causes the tertiary structure of proteins to occur? So the factors that causes tertiary structure of proteins to occur are the following: covalent bond, especially for the for the disulfide linkages between cysteine side chain, the hydrogen bonding for polar molecules, okay, the salt bridges between our charged amino acid, for example, histidine, lysine, aspartic acid, glutamic acid. So their interactions can form salt bridges, and the hydrophobic interaction of your nonpolar residues. So all of this play form, no, in determining the tertiary structure of the protein. Okay? So dito natin malalaman kung siya ba ay globular, okay? Ano yung itsura niya as a whole, okay? So lahat ng mga sinabi ko kanina na factors affecting the primary, secondary levels of structure encompassed sila dito sa tertiary structure. Okay? So these are the reasons why they are stabilized. Okay? So ito, so this is a good summary picture of all the forces that stabilize the tertiary structure. You have your hydrogen bonding here and here. These are the salt bridges between charged amino acid residues. Okay, so this is the hydrophobic interaction between your nonpolar residues. This is the disulfide linkage of your cysteine, and this is the hydrogen bonding again. Okay, and for the quaternary structure, hindi na to masyado discuss in depth. So even sa amin sa masters, di na rin yan diniscuss masyado. Okay. Ang quaternary structure lang is how all these protein tertiary structures work as a whole. Okay. So for example, human hemoglobin is a quaternary form yan. Okay? Quaternary structure meron siya because you have 4 different uh, proteins na nandun. Okay? So 4 different proteins yung associated in one hemoglobin molecule. Okay? So inside them, meron tayong iron containing heme unit. Okay? So this is the quaternary structure of your hemoglobin. So you have your structure here, structure there, structure here, and the structure here. Okay. So as you can see, each of them contains heme group, and that heme group is the primary reason for the absorption of oxygen in the blood. Okay. So when our oxygen attaches to our heme group, no, yung quaternary structure niya, nagbabago siya ng conformation. Okay? So, dalawa yung conformation ng ating hemoglobin. Wala dito. So, you have your relax and taut state. No? Okay, so let me show you a picture. Hemoglobin, relax, and taut. Okay, so ito yung conformation na nangyayari sa quaternary structure ng ating hemoglobin when oxygen attaches to it. Okay, so for the taut, ito yung may nakabind sa kanya, and then the relax, yun yung magre-release siya ng nakabind sa kanya. Okay, so pwede yan mag-contract or mag-expand. Okay, so that is the taut and the relaxed state. Okay, so yan yung yan lang yung function ng quaternary structure in terms of hemoglobin. Okay, so yan. So wala naman masyadong dyan, wala naman masyadong ganap. Okay. So this is the heme molecule that is in the subunits of our hemoglobin. Okay, and this is the iron. That's why kulay pula ang ating dugo, and lasa siyang bakal. Ayan. And then so integral membranes are also forming quaternary structure. Ano kaya itong protein na to? Walang nilagay dito na pangalan. Okay? So this one as well, I think ito yung pinakita ko kanina na nasan yun? I think ito yun sa Pseudomonas aeruginosa. Parang ito yun, di ba? Yeah, so they are similar. Okay, so let's talk about denaturation of protein. Okay, so last part na tayo. So when we say denaturation, ibig sabihin nasisira yung protein. Okay, what happens when our proteins get denatured is that the tertiary structure and the quaternary structure gets destroyed. Okay, so nasisira yung tertiary and quaternary structure. So ano mangyayari? So let's say for example yung itlog, no? So pakita ko sa inyo yung albumin. Hanapin ko muna dito. Albumin. Chicken albumin, kasi ito ginagawa natin sa bahay. We are familiar with this one. So this is albumin, okay? Ayan. So as you can see, I think this is a dimeric protein. Dalawa yung groups niya. So you have this group here and another group here. And as you can see, for each group, you have your beta sheets and your alpha helices. Okay? So, anong nangyayari when our, let's say, yung ating albumin sa egg, no? When they get denatured, namumuti siya, no? Bakit kaya, no? Ba't siya nagiging solid, no? So, this one is liquidy. This is globular. Pero bakit kapag ininit natin siya, namumuti siya? So, what happens here is that ganito daw. Okay? In the denaturation of protein, okay, you are destroying the tertiary level, the tertiary structure, okay, of your proteins. So, bale, kunwari yan naka-fold ng ganyan. So, when you react it with some denaturing agents, no, such as mga detergents, okay, heat, and then pH, ayan. So, mag-uncoil sila. So, when they uncoil, ayun, dun siya mag-start mamuo, okay. Because when they are uncoiled, hindi na siya nagpo-form ng globular structure, no? So most likely, naging flat na lang siya, and that flatness causes them to solidify. Okay? So yun, nasisira yung tertiary level ng structure niya. Okay? And then sabi dito, one thing is interesting na sinabi dito is that denaturations are reversible. So ibig sabihin, yes, it is indeed possible to convert cooked egg back to the uncooked state. I think nakita nyo yan sa Facebook no nakaraan. No? So yes, it is true. Possible yan, yung luto na itlog, ibalik mo siya sa original form. Pero, hindi ganun ka-sure no, kung 100% na functional pa rin yan. Okay? So that's the idea. Denaturing agents, so you have your heat, no? So when heat are introduced to your proteins, no, ang nangyayari is that the hydrogen bonds, no, can be disrupted, no. So just like in water, no, the reason why water boils is that the hydrogen bonding gets disturbed, okay? So when the hydrogen bonds get disturbed, ayun, magbo-boil na siya. So sa protein ganun din. So when you cut your hydrogen bonding, so itong mga coils natin and even the beta sheet, they can uncoil, okay? So the uncoiling causes them to solidify. We can also use urea, okay? So urea is also used to disrupt hydrogen bonding because this can form hydrogen bonds as well. So instead na yung protein natin mag-bond with each other, they will instead bond to urea. Surface active reagents such as detergents, okay? So sila ay ginagamit din for disruption of hydrogen bonding. Reducing agents, they are used to reduce the disulfide linkages. Okay, the disulfide linkage results in the tertiary structure of protein kasi. So if you cut that disulfide linkage, yun, mawawala na yung tertiary structure ng protein natin. Heavy metal ion, this is why heavy metals are toxic to humans because we have proteins and apparently our proteins can interact with that, especially the charged groups, okay? The aspartic acid groups and the glutamic acid groups. No? So when they interact with heavy metals such as cadmium, lead, mercury too, okay? So yan, instead na alpha helix semaform, mag-clump siya. So that causes destruction of protein, which can lead to mutation and worst case is the cancer. Okay, so yeah, and then alcohol can be also used to denature our proteins. Okay, so yeah, for say maybe for the same reason that it can disrupt hydrogen bonding. Okay, so those are the reasons why our proteins get denatured when you heated them. No, yung hydrogen bonding napuputol, and that causes them to unfold. And then when they unfold, yan. So we have here a picture, ano to, ribonuclease. So for example, we have here the ribonuclease. Okay. So this is your native ribonuclease daw. So this is one of the protein that is used in saan ba to ginagamit? Siguro sa sa DNA sequencing na. So bale, ginawa daw dito ng mga scientists that they reacted this with urea and beta mercap mercaptoethanol. Okay, so ang purpose ng mercaptoethanol is to cut the disulfide linkage. Ito yon. And the purpose of urea is to uncoil them. Okay, so that the hydrogen bondings will be disrupted. Okay, so the beta mercaptoethanol, puputulin niya yan. So that causes it to uncoil. However, when you remove this, okay, and you oxidize this in the air, mangyari, magko-connect uli sila together, producing the same uh, protein. Okay? So, ibig sabihin, yes, denaturation is reversible, but maybe for the context of itlog na nakita nyo dati sa Facebook, I don't know if masarap pa rin yun. Kasi, ano eh, kung ano-ano na hinalo nila sa egg. Pero yeah, denaturation is possible. And I don't know if the albumin there still works. Okay? So, yun, we ended our discussion quickly. So anyway, so we're done with the discussion on protein. Okay? So, for the rest of the session, magpapakita na lang ako ng iba't ibang protein siguro. And then I want you to tell me the conformations. Sige, we have a question from Kelvin.
Sir, paano po perform yung double helix ng DNA?
Now?
Ng DNA po, paano po perform yung double helix?
Yung double helix naman na DNA. Okay, so that's based on the hydrogen bonding naman between your, instead of amino acid, no, you use your nucleotide bases. Okay, so very different yon. Okay, with our proteins. Okay, kasi in the proteins, the reason why we have double helix dito sa proteins is because of every after three residues you have the same polarity. So when they match, they coil. Okay, in the context of DNA, no, yung kanilang mga residues ay limited lang to 4. Okay, 4 for humans. Okay, the adenine, thymine, guanine, cytosine. Okay, so medyo analogous siya with kung ano meron tayo dito sa protein. Medyo analogous pero technically not the same. Okay, so ang sagot doon bakit nagpo-form ng double helix yung ating yung ating DNA is that number one, hydrogen bonding is observed between your nucleotide bases na the ATGC. Okay. And number two, okay, just like the proteins, no, the structure of your nucleic acids, no, they can also form hydrogen bonding with each other. Let me show you yung ating DNA. Nucleic acid. Hindi kasi natin madidiscuss nucleic acid dito eh. Sayang. Maganda din yun. Pero wala eh. Nakapag na ba kayo? Something like this na topic dati. Mga cytogen, something like that. Okay. So, hanap ako magandang picture para mas makita nyo yung hydrogen bonding between them. Upuan ko yan. Tumutunog eh. Ayan. So, this is the reason why we have double helix. Okay, so your adenine, thymine, no? At the end of their chains, you have your hydrogen bonds. Guanine, cytosine, tatlo yung kanilang hydrogen bonds. So, kapag marami kang guanine, cytosine, mas matibay DNA mo kaysa sa adenine, thymine because guanine, cytosine, tatlo yung hydrogen bonds niya, adenine, thymine, dalawa lang. Okay, so yan. And itong chain natin sa backbone, okay, so this phosphate chain here, no, so they can also form hydrogen bonds, no, kasi may sugars din dun eh. Okay. So yan. So yan, itong mga hydroxides doon. Sorry, hindi pala hydroxides. So itong mga phosphates natin na to. Okay. Hanap ako ng magandang picture talaga. So mahirap maghanap ng picture eh. So wala din akong copy dito ng PowerPoint na to. Pero yun yung idea, no, ba't siya nagko-coil? Due to hydrogen bonding. And then Yan. So pag nag-hydrogen bond sila, they form ladder, and then gagawin na lang ng histones. Okay? Histone is a protein that causes them to coil. Okay? So yung histone is squeeze siya para maging ganyan. Okay? So parang ladder siya, and then some proteins like histones, yan, tutulungan niya yan mag-coil like that. Okay? So yun yung sagot ko dun. So sayang wala akong magandang pictures eh. Wala akong magandang picture niyan kasi kailan ko ba last na tinuro to? Tinuro ko to last year pa. Last year pa, kaya hindi ko mahanap din yung file. Okay, so yun yung reason din. Ba't siya nagko-coil na? Hydrogen bonding din. Okay? Okay ba tayo dun, Kevin? Kelvin?
Opo, sir.
Or, sige. So, hydrogen bonding lang din yung kasagutan sa lahat. Actually, lahat ng bagay sa katawan, hydrogen bonding yung mga may resulta niyan. Okay? So, ito. So, I will show you some proteins, and then you need to tell me kung asan yung kanyang mga nasan yung kanyang mga secondary structures. Okay? So, kunin ko yung COVID-19. Okay. So, this is the main protease of COVID-19. Okay. So, what secondary structures can we see here? Okay. Ano yung mga secondary structures na nakikita natin? Okay. So, could you enumerate it? Pwede kayo mag-recite no? Open your mic. Kasi titingin ako sa camera. Ay, tingin ako dito sa second screen. So, anong secondary structures na nakikita nyo? You have your... Okay, so Kelvin, ano meron dito?
Meron po alpha helix.
So, alpha helix. So, ito yon. Okay. Ano pa?
Tsaka po, beta sheet.
Okay, so beta sheet. Ito yan. Ano pa? Meron pang isa.
Yung random coil, sir.
Ay, so the random coil. So usually the random coils connect your alpha and beta configurations. Pwedeng alpha-alpha, pwedeng beta-beta. So as you can see here dito sa COVID-19 protease na to, dito natin makikita yung alpha-alpha motif. Okay? So for example, ang hirap kumanap ng angulo. Ayan. So this is alpha, and that is connected to this alpha chain here. Okay? So that's the alpha-alpha unit. Okay? So is this a globular protein? Maybe it's the globular. Kung gusto niyo makita kung globular yan, ganito gamitin yan. So you select the add, then you choose protein. So you add selection, protein yan. Then piliin mo yung Gaussian surface. Upuan ko yan. Ayan, create component. Yan. So this is how it looks like. Okay? Ayan. So, di ba yun nga? Yun sinasabi natin na okay. Globular siya. Okay? Kasi nakapabilog siya. Okay? So, yan. So, I will look for another protein. Teka lang. Anong protein kaya? Let's say hexokinase. Okay, so we have here the human hexokinase. Ayan. We still have 7 minutes. So, for the human hexokinase, okay, ito yung first enzyme na ginagamit in our glycolysis. Ayan. So, this is your hexokinase. It appears to be dimeric protein. Dalawang structure meron tayo dito. So, the orange one and the green one. So, as you can see, marami siyang alpha. Puro alpha actually. And then some onting beta. So, beta, especially in these regions, yan ang beta. And then you have your random coils, which connects the beta or the alpha. Okay. So, let's try to see if we can also do we can also perform the same thing. Tulad ng ginawa ko kanina. Gaussian surface. Pag ito nag-lag, ayun, kala ko maglalag siya. Okay. So this is our hexokinase. Okay, the thing with enzyme is that may nakikita kayong portion na empty, especially this region here. So next meeting, ito yung discuss natin, enzymes. Okay. So this region here is called the active site. Okay. And the active site is the position where your residues can actually enter, the substrates can enter. Okay. So dito nangyayari yung acid base, dito nangyayari yung enzyme catalysis. Okay. So kapag yung ating glucose pumasok dito, madadagdagan siya ng phosphate and ilalabas siya. Okay? So, these clefts here in your protein, they are primarily called the active site. Okay? Anyway, pag nag-discuss tayo ng enzymes next meeting, mas makikita nyo yan. Okay? Let's check the polarity of the surface of the molecule. Sige. Let's check the polarity. Mag-a-add ako. So, dito sa Gaussian surface, magdadagdag ako ng polarity. Tingnan ko kung kaya ng computer ko. So, I will check kung kaya ng computer ko. Hindi niya kinaya. Sayang. Okay. So, hindi niya kinaya. Anyway, pwede mo kasi makita dito yung yung mismong surface ng protein mo. Okay. So, let's try 1 RTQ na lang. We have 4 minutes pa naman. So, pakita ko sa inyo yung itsura ng surface ng proteins. Okay. So, dito natin mapapatunayan yung sinasabi ko kanina. Okay. Na yun, yung polarities iba-iba. Wala, hindi na siya nag-load. Ayaw na niya. Ayun. Ayan, so ayan, we have the hydrophobicity. So ano meaning ng hydrophobicity? Yung kung polar ba ay nasa labas na. So as you can see, although maraming non-polar, no, it is important to know that may mga non-polar residues outside. Okay? So for example, ano kaya to? Okay. This is glutamic acid here. Okay. TRP, tryptophan, if I'm not mistaken. Phenylalanine. Glutamic acid again. Okay, lysine. So makita mo, in the pockets of your surface of your protein, you have the polar residues outside it. Okay? So yun yung sinasabi ko kanina that yung ating protein in order for it to be globular, no, the non-polars are usually embedded inside and the non-polars are sticking outside. No? And yung nakikita yung red dots, okay, those are actually water molecules. So as you can see, in the colored regions kung saan may polar tayo, doon mo makikita yung karamihan ng water molecules. Okay? So that causes them to be soluble in water. Okay. So there are many things that you can do out of this website. That's RCSB, Protein Database, Protein Data Bank. Okay. And I highly suggest you to learn more of this. Okay. And I think ituturo naman to sa inyong higher biology subjects. Okay. So that ends our proteins. No, hope supposedly dapat magku-quiz kayo pero I will continue with the enzymes na lang next meeting. Enzyme is one session lang naman. And then next week, wala na tayong klase so that you can comply with the requirements. So, quiz lang naman yung kailangan dito. So, next week na lang, okay? So that you have more control of your time. Okay? So, Thursday, enzymes tayo. Okay? Isang meeting lang yan eh. So that ends our session. Thank you, and I hope you learned something today. So I'll see you again next Thursday, and good luck sa upcoming exams. Bye bye. I'll upload this video on YouTube now. Okay, bye bye po.